And of course, thanks to the organizers and of course to the Paul G. Allen Foundation for the award that we've been working on. So today I'd like to tell you about a snapshot of some of the things we're doing in this new area that we're referring to as active genetics, where the work is based on self-propagating genetic elements. And this work was done initiated by Valentino Ganz, who played the key role in getting it all started, and almost everything I'll talk about today comes from that. And in fact, this artwork is from him as well, because he's an artist as well as anything else. And the Vitruvian mosquito over here is one of his, I think, cooler pieces of work. So this is Valentino, and I'm sure by now everybody is quite well aware of how CRISPR works, so I'll just be very succinct about it. You have two components, an enzyme, Cas9, that doesn't do anything much on its own, but when it binds a guide RNA, shown in green here, it can target that Cas9 protein to pretty much anywhere you want to go in the genome with a slight constraint of a PAM sequence, and then it cuts at a very defined location. And so you can cut the DNA almost everywhere, anywhere you want in the genome with this. It's the miracle of our time. And so if you have this sort of targeted molecular scissors and you cut one of the two chromosomes you have, one from mom, one from dad, there are several possible outcomes. And this is a gross oversimplification, so for those who want a little more discussion about this later, I'm happy to do it. But if you want to repair that gap, you have to, or the cell will die. There are largely two ways of doing it. One, which is ultimately has an outcome of error proneness, which is to just fill the gap with some kind of like bubble gum to just repair it. So the indels, little short insertions or deletions of material, and that just sutures the chromosome back together and you can go on. And this happens throughout the cell cycle. It's a very common thing in somatic cells. The other is homology-directed repair. It also happens in somatic cells following replication, but as it turns out, and again, I'll be happy to go over this more subsequently, it is, by good fortune for our work, almost the exclusive pathway that's used during meiosis for germ cell production. And so these are the two alternative pathways, and if you use the HDR pathway, what you do is you copy information from the intact chromosome into the gap and repair it. And so that, as it turns out, is a very dominant or probably exclusive pathway in the meiotic lineage. So what Valentino did as an idea originally was he made a very simple DNA construct in which he had the Cas9 and the guide RNA encoded in such a way that they be produced in the germline, and in his case in other cells as well. And then he flanked them with DNA sequences from the genome, homology arms, these HAs, that exactly flanked the cut site. So the idea was that if you made a cut, the cut would be repaired by this little pseudo chromosome by copying in the genetic material that it carried into the cut. And then when that happened, it's on one chromosome, you can imagine, like mom's chromosome, let's say. It makes the protein again and the RNA, and then it cuts exactly at the same site that's opposite to the place where the cassette got inserted. And now it's the same thing, except the homology extends out across the whole chromosomes, and this time it really is not tricking it into thinking it's a chromosome. It really is a chromosome. And again, it copies itself. And so now all of a sudden we're biallelic for it. Instead of having just one copy from mom that's one thing and one copy from dad that's something else, both copies are the same thing. Both copies carry the element. And so this is this little diagram or this little animation of that. The Cas9 gets made. The normal fly starts off being brown. This is a gene that codes pigment, so if you cut it, it makes it zip, go yellow. And so that's what happened. His flies all turned yellow. And I have to tell you, just as a scientist, I've been a geneticist all my life, and when he came in and he showed me a vial of flies that normally would have been heterozygous, one good copy of yellow, one bad copy of yellow, standard recessive mutation, and all of those little puppies were yellow, I could not believe it. So anyway, that gave rise to this general idea and field that we're trying to refer to as active genetics more broadly, because there are lots of different applications of this kind of technology. And I'm just going to go through by stint of time limitations a few of these possible things. The most obvious are called gene drives, which is a way of dispersing genetic information through a population. And I'll talk about that first, and then I'll get to some of these other elements. And chasers, which are elements that can work together with drives, and I'll talk about that in sequence. So first the drives and then the chasers. So this is Tony James, the person we collaborated with on the gene drive work. He has spent his entire career trying to create and successfully creating molecular tools in mosquitoes to bring it into the modern era of molecular genetics, and has pioneered transgenesis in that species, as well as devoting himself to defining gene cassettes which, when expressed, can render the mosquito immune to the propagation of malarial parasites, so that they no longer can propagate malarial parasites once they've bitten a person that carries the disease. And after years of his work, really, this summarizes, sadly, in this very quick diagram, about 20 years of Tony's work. He created a very sophisticated genetic element that contains cis-regulatory elements normally present in the genes that are involved in digesting blood and respond to a blood meal. Only females bite, males don't. So this is a gene that would be active in females, and he attached it to an effector molecule, which are single chain antibodies that bind to the parasite. So they're genetically encoded antibodies, basically, and they bind to the parasite and they essentially prevent its propagation. So it's like genetically immunizing the mosquito. And at the end of the work where he characterized these, it gave 100% inhibition of propagation of the malarial parasites. He said if this were coupled with a mechanism for gene spread, then it could create a sustainable malarial tool, a tool for sustainable disease control. And so, that was exactly when, we read this paper exactly when we did these, when Valentino got these results. And so we called him up and said, well, maybe we could hook our widget to yours and see what goes on there. And to make a long story short, it, was remarkably effective in being transmitted to 99.5% of the progeny. And so that then, in principle, you could take a genetic element like that and bring it into a population at 1% of the normal mosquito populations. And to put that in context, standard sterile insect technology often puts in 10 times as many as are present. So this is like 1/1000th as many mosquitoes as would be used for sterile insect technology. And then within one season, 10 generations, it could, in principle, go through logistic growth to fixation into the population. And so I don't want to go into all the various little accessory elements that you can put in principle into this and to remove it and so forth, which is a talk in itself. But just suffice it to say that you might imagine that some of those anti-parasite molecules, would get, the parasite would get resistant to them, and you might have to come in and update the genetic element. So that we have these genetic updating elements that we call chasers, and these are just backronyms, so don't worry about all of this. And we hear so many acronyms anyway, but it's chasing along after the drive element. That's the idea, it chases. Okay. And so the idea here is that it only carries the guide RNA at the site of insertion into the genome, but not the Cas9. So it's not like a car where the gas pedal is stapled to the floor, but rather a car where you can change, you can depress or not the gas pedal being Cas9. And so the idea here then is that this would be an example of such a DNA element before it's been inserted, and they'd have a couple different guides in it, as well as uptake cassettes that could say have some new anti-parasite molecules, so that in case they had gotten resistant, then this could drive through. And the idea then is that this guide RNA here, for example, one would be the one, like in Valentino's experiment, that inserted it into a given location. And in the presence of Cas9, the Cas9 that could be provided either in trans by a standard Mendelian source of Cas9, and I'll get to that in a bit, or if it were provided by a gene drive, which is propagating itself throughout the population, then piggybacking on this or chasing after it would be this element that uses the Cas9 from the gene drive to combine with this guide RNA to drive this element into this location. But then the other guide RNAs that could be on there could be used for other purposes. So for example, guide RNA1 could be used to disable a host factor that's known to be necessary for the propagation of the parasite. Guide RNA3 could be used to do something that I'll focus on a little bit here, which is to target a known, and there are many of these examples, a known point mutation in an ion channel that renders the mosquito resistant to pesticides. This is a huge problem in the field of mosquitoes control. Some mosquitoes are resistant to as many as 3 different pesticides. So the idea is to have this thing called, let me just go back on that if I can. is to have this idea of allelic drive, wherein what you do is you have a guide RNA that cuts the resistant form of the ion channel, but not the sensitive form. Now, if that's created, as it would logically be, in a sensitive strain of mosquitoes, and that is a mosquito that would be sensitive to the insecticide, then if that insect mates with one that has the resistant allele would be cut because the guide RNA just cuts the resistant one but not the wild type version, and then the repair would be from the wild type version, so that would render the mosquito in principle resensitized to the insecticide. That's the concept. So as a sort of test case for this, we use in fruit flies the notch locus, which is a locus that has a lot of advantages. It's very well studied. There are loss of function and gain of function alleles of it, so we can distinguish all these various genotypes. Loss of function alleles in heterozygosis cause thick veins. The abrupt X dominant allele causes the opposite phenotype of loss of veins. Homozygous loss of function notch mutants are dead. Homozygous gain of function mutants are viable. So those are the kind of rules, and I realize that's a lot to hold in your mind, but I will nonetheless come back to that. But the important thing is two things. One, they have opposite phenotypes, loss of function, gain of function. Gain of function is cool if it's homozygous. Loss of function is not. Okay? So what we found, or Annabelle Guichard, the postdoc who did this in the lab, what she found is a situation where there was where the wild type allele was subject to cleavage because it had a PAM site, but the abrupt X allele, the gain of function allele, did not. So it lacked this necessary sequence, which again, it's not a huge constraint on the cutting, but you can make use of it by finding an allele that does or doesn't have that sequence. So the bottom line is that the abrupt X allele is uncuttable in this case, whereas the wild type allele is cuttable. And so the experiment then is you take a gene drive element that's got a guide RNA that propagates the element itself. This little clicker thing is running out of steam here, but the guide RNA in brown is the one that propagates the element to its location in the yellow gene, the same as Valentino's gene drive thing, but it doesn't have the Cas9 in this case because we're trying to use that in a conditional way. And the guide RNA, too, cuts the wild type notch allele, but not the abrupt X allele. So it can cleave this one, but not this one. Okay? And so then what happens is we then ask, well, if you do, and you cross that to males that carry a wild type allele, what happens? And you can just score that in both males and females. And so if you score it in males, you ask, do you get 50% of those that have a wild type allele, 50% of those that have an abrupt X allele? That would be what you would expect if you had nothing happen, if it was just standard Mendelian inheritance. Or do you get a disproportionate fraction of them that inherit the abrupt X allele? And the answer was that 94% of them got the abrupt X allele, which is quite notable, and only 6% got the wild type allele. And when we looked in females, the numbers weren't all that different. And that was a bit of a surprise because in females, what we'd expected is you'd be able to get another category, which is the notch minuses over plus, because notch is just a recessive mutation. So you should be able to get notch minus over plus. So we got numbers instead that were much more just like what we got in males. And so we wondered what was going on here. And one idea was, well, if there's no notch minuses, maybe what's going on is that if you inherit a notch minus chromosome, then that would be over here, then the other one that you're getting from the male, maybe it's also getting cleaved by the Cas9 and the guide RNA that were present in the egg of this mom over here and is being inherited not genetically but by perdurance of the Cas9 guide RNA, which we call shadow drive. It only works for one generation. And if that were true, if that explanation for that were true, then we call that lethal mosaicism, so that the body has, all of the cells have one bad copy, and some of the cells have two bad copies. And if enough cells have two bad copies, you're croaked. That's the theory. That's the hypothesis, okay? And to test that hypothesis, then we thought, okay, then if that's what's going on, you've got this lethal mosaicism, then that depends on that allele being mutable. So if we then instead we change to cross it to a notch abrupt X allele where that is no longer cleavable, that is an allele that's resistant to the guide RNA, then you shouldn't get lethal mosaicism. And indeed, we got the same thing, basically the same percentage of males and so forth. And then in the case of females, now we got the other categories about in the same percentage again, but importantly, we did find notch minus over notch abrupt X alleles. Now, it turns out that that particular genetic combination isn't entirely viable, isn't so happy. It lives, but you have to kind of hunt for it. But you can actually, we were actually able to get a much more precise estimate of it by looking at embryos where we get everybody, and it happens about 10% of the time. So it's kind of what we expect. And the thing that's very interesting about that in terms of a prognosis for using this in the field for going after things like insecticide resistance is the sodium ion channel is exactly like the notch gene. It's an essential gene and would be subject to lethal mosaicism. So not only if you have, it's one of these things where everything conspires in your favor instead of against you. So first of all, the drive thing works pretty well, which is cool in itself. But in addition, when it doesn't, it tends through lethal mosaicism to kill the animals that have the mistakes. So the only thing you inherit, the only thing that comes through is what you want. I mean, really, is life like that normally? Not usually for me anyway. Okay, now I'd like to then shift to using these elements in a more regulated way to, again, with a Mendelian source of Cas9, that is an accelerator that can go up or down, and we call those CopyCat elements. And again, what happens is that they've got the guide RNA where they're inserted and they can cut the other allele, but the idea is that, that then you can control it by crossing the Cas9 out. So again, with this idea of sort of, the first speaker was speaking very convincingly, I think, about the need for humanized models in mice and other things. So imagine that we wanted to make several different kinds of humanized models of mice. Those that had a humanized immune system, those that were humanized for drug metabolism, those that were humanized, say, for as an Alzheimer's model. Okay, so now imagine that you have, each of those mice is a complex genetic mix of things. Okay, how would you combine them in a modular way? And this offers, I think, active genetics offers an approach to that. So here what we have is the Cas9 is indicated by the circle. These are the self-propagating genetic elements in the presence of Cas9. And so if you cross two mice that have such things, and then in their progeny, they each inherit one of these elements from each parent, but they're self-copying, so in the presence of Cas9. So all the kids get both of them instead of just 50%. And so now all the offspring look like this. And this is theoretical. We haven't done it in mice yet. Those experiments are going on, but we haven't got that data. So this is just theory. And then you cross it to another complex model. Let's say that's the humanized mouse for the immune system. And again, what would happen is that the progeny should be heterozygous for each, but then in the presence of Cas9, they would copy. And so all the kids would have these. And then you take two such animals and you cross them to each other if you want to just have a stable line and go back to normal things. And then you just remove the Cas9. You select the animals that get this non-Cas9 chromosome over here and this non-Cas9 chromosome over here, and then boom. And then you can repeat this with the model for Alzheimer's or some other model, cancer model. So I'd just like to end by giving one story, which I mentioned very briefly at the end of my last presentation at the Allen Foundation, but we've gone a little further, so I'd just like to revisit that. I mean, let me just go back here. This is something we've been studying for a long time in the lab, the generation of this vein. And it's a very esoteric thing, I admit, but it is actually this study from which Valentino got his idea to do everything. So it's one of those examples of just allowing people to do their thing in basic science can get you forward. But anyway, we want to know how that L2 vein forms, and I don't want to impede you with it, but here's the molecular expression of this gene CNRPS that is the precursor in this stripe to make the vein. And there's a lot of work we've done in the lab that says this expression here is what makes that vein, and it's right anterior to this pink domain. And that's just stuff that, again, has come from our lab and many other labs that are trying to work out this gene regulatory network for the wing. And so the idea is that we know where the cis regulatory element is that creates that stripe of green. And so what Valentino did is he made a CopyCat element that has two guide RNAs that delete that enhancer element. And when they delete the enhancer element, there's no L2 vein. And I don't want to go through this in extensively here, but just to show that if you combine that with a gene drive element, that the majority of progeny that get it in different crosses copy the element. In 4 of the crosses, it copied to 100%. In 3 of the crosses, it copied to 90% roughly, and in 2 crosses, it failed, and that's an interesting thing that we can talk about if people want to. But it worked pretty well as the bottom line, and because it worked so efficiently, what was possible to do is to use it to bring in the genetic element from another species. So we take out the fruit fly one and replace it with one from another species. And so obviously, if you just replace it with the one from Drosophila, you get the expected result of where the vein comes back. But then you can go to a more distantly related fly that's about 40 million years or so away that has it's a much bigger wing, but it has the vein in roughly the same location, and it restores the formation of the vein perfectly. So this is lacking the Drosophila melanogaster cis-regulatory element, but in its place has this other one. And so again, you can do that with the housefly or with the housefly, and in that case, what you see is that the vein comes back, but it's not at the right location. It's anteriorly displaced, as is the vein in the actual housefly. And the same thing with the medfly. The rescue is not as complete, but again, the piece that's rescued is more anteriorly displaced as it is in the other organism. And so in the case of the housefly, which had the most dramatic shift, and the whole vein, we looked at by in situ hybridization at what happened when we put that piece of DNA back in to the Knirp's expression pattern. And what we found is that it got significantly broadened. So it does bring the expression back, and it brings it back in a broadened pattern. And previous work we'd done had shown that if you provide low-level ubiquitous expression of Knirp's, it tends to shift the vein anteriorly. So it provides an adequate explanation for the phenotype that we see. And so I'd just like to end by pointing out that over the years, and very importantly, with the very generous support from the Allen Foundation, we've been able to put a team together at UCSD that is exploring active genetics, not just in mosquitoes and fruit flies, but for human therapy, mice, fish, plants, and yeast. And hopefully soon we'll have results in some of those other systems that we'll ultimately be able to share, seeing if this kind of systems work in other organisms. My prediction is it should work in every genetically in every organism that has genetic recombination, sexual recombination. Every organism of that kind, in principle, I believe this will work in. And the reason is because homology-directed repair is a commonality of the germline of all such organisms. And again, I'd just like to end by recognizing the people. Valentino Gantz, he's the guy who started the whole thing. Shannon Zhu has been working very closely with them. She's the one who made all those deletion studies that were for the fly wings. Annabelle Guichard is the one who did the allelic drive experiments. And then we have had lots of colleagues and collaborators at UCSD and at other locations. And of course, thanks again for the funding from the Allen Foundation. Thank you.
On the way, so it's a great talk. So allelic heterogeneity, or allelic drive. So it strikes me that the potential challenge there would be allelic heterogeneity with respect to resistance mutations. So, and this is something I don't know enough about. Like, do the mutations tend to be stereotypically clustered, or one mutation that dominates in a population that you could target, or is it?
Right. So there are a couple of answers to that. So the particular mutation, the 1014 L to F mutation, is something that you see over and over and over again in response to DDT or pyrethroid pesticides. So it's a ubiquitous and very common mutation that is a leading cause of insecticide resistance. That said, there are about 10 or 20 other important mutants that do that too. Some of them are also in the same gene, the sodium ion channel gene, so there are other ways of thinking about that. Another way is, instead of just changing the allele, is to change the whole section of the gene with a recoded part of the gene that doesn't have the insecticide-resistant parts. I call that a snowplow. That's another way. But basically, what you'd have to do is you'd have to go into each population and see what are the resistance parameters there and go for it. But you're absolutely right. There's a lot more. This is just a general concept at this point. But the idea that you could drive, the other thing that's the other part of your question, though, is that it turns out from studies in Africa with people who've been going into homes that have bed nets and so forth, where pesticide resistance is a big problem. It turns out that the quantitative analysis of that suggests that if you can just have about 30 or 40% of the insects be sensitive to the pesticide, that you can control them. So in field studies suggest that a modest increment in their sensitivity to the pesticide could have a huge effect. Pesticides cost about $200 million to go from development to the field. So if you could then, and there hasn't been a good new one that in the last 10 years it's been developed. So if you could use this kind of technology to help with that, then not only do you have gene drive as a way potentially to go after the parasite, but then you give the standard vector control people some leverage on their standard control methods.
Last question?
Yeah, the terrific work. It's always remarkable just to see the amount of recombination that goes on, especially with like a 17 kb piece of DNA. I was wondering, have you used sort of the power of the Drosophila genetics to actually try to sort of break that and find out what are the key players in recombination? Say something that would only integrate a very small piece of DNA, but not the larger ones and things like that. Because it seems like there's something that potentially to be learned also from just basic recombination.
Oh no, absolutely. I mean, that's an excellent question, and of course we're fascinated with doing that. And we're making all kinds of genetic elements to try to parse the mechanisms by which it works and the effects on recombination machinery. Also, we're trying to look at how well it copies somatically versus through the germline. We know it copies quite well through the germline, but we don't know for sure. But our suspicion is it doesn't do so well through the somatic cells, which is why sometimes we think it fails because it gets spuriously processed in somatic cells before it gets into the germline. And so we're making counters. A copy counter is the next one we've got that's going in that will only fluoresce when a copying event has taken place. And so we're going to try to answer questions like that and then do genome-wide screens for those that increase or decrease the frequencies of those things. But you're right, the size thing is a very interesting one and has very important implications for genome engineering down the road.
Thank you.